Hello?
Yes, we can.
Oh,
Yes.
okay. Okay, so first of all, thank you so much for having me and bring me here to give to share my story about Manos binding lectin MBL. So I wasn't sure what to do, but so I decided to give kind of broad sort of background what MBL and my thinking about MBL at this stage. So at this point, MBL to me, it's multifunctional. I hope I can convince you what I'm telling you is true. And MBL is like sentinel, so providing constant surveillance through, and also kind of house cleaning, and also through coagulation. I'd like to call it bionic glue. And lastly, it is a disease modifier. So as long as you don't have anything, you may not feel it, or you wouldn't know till you look for it. But if you have underlying problem or some infection, then MBL deficiency in particular would make you feel something wrong. Now, I'd like to start with a big picture, at least to me. So thanks to host defense system, we have innate immunity, which kicks in second or minutes really after our body is exposed to danger, could be just injury or invading pathogen, and provide local containment. And one of this system is called the pattern recognition molecule, which MBL belongs to. And if we now this sorry, oops, sorry, so oops, so I'm messing about. Okay. Now, innate immunity has linked to acquired immunity. Now, which takes at least a week, a long time, but provide systemic and target-specific protection, often quite for lifetime, since it has memory component in it. Now, what is MBL? It belongs to the collectin family, and as I mentioned, pattern recognition molecule of innate immunity. By itself, functions as opsonin, which facilitates phagocytosis by so-called professional phagocytes like a neutrophil and macrophage. And also in cells, had served as a post-translational glycosylation, so located at border of ER and Golgi, which was actually done by the other research group. And also MBL in complexes with a mass, MBL associated stem proteases activate the lectin complement pathway and also function like thrombin. Now, I mentioned innate immunity target is broad. So when we started MBL target, we really thought only pathogen. So bacteria, fungus, and virus. But we now know MBL can recognize much broader sugars, and ourselves, like once it's injured or transformed like a cancer, MBL can recognize also some enzymes and amyloid beta, like a Alzheimer protein. Now, as a function, by itself can serve as opsonin, so increase phagocytosis by neutrophils and macrophages of MBL target. And in a complex with MASP1, MASP3, so MBL's functional MBL is hexamer. So then this complex activates the lectin pathway. So halfway through alternate pathway as well as classical pathway. Now, our contribution to this was this MASP1 MBL complex can function like thrombin, meaning transformed from fibrinogen, soluble form to fibrin clot. And the MDL is primarily synthesized in the liver, but some small portion expressed in small intestinal mucosal epithelial cells. And I personally believe it should influence on the microbiome. We heard earlier this morning as well as yesterday. And even though MBL is synthesized in the liver, most circulates in the blood and seeps into tissues. So far, MBL has been identified in CSF and alveolar lavage and even in the skin. Now, I'd like to talk about the molecular structure of MBL. So starting from 5', it's so-called system-rich domain, which we think, well, actually it is, to bundle monomer of MBL, and followed by collagen, long collagen region. Hence, it is called the collecting. And CRD, carbohydrate recognition domain, which is primary target recognition domain. And MBL has 4 exons, and of which first exon, towards 3' of first exon, the known SNP, they're called D, B, or C allele. So if you have any of these, MBL secretion is reduced. And in addition to this, at least 3, actually we know more, 5' open reading region as well as the 3'. And because of these alleles, we have so many haplotypes. This ended up in broad range of plasma MBL levels from undetectable to, we've seen 20 microgram per ml, which is a lot. But average, if you are wild type for both alleles, 2 microgram per ml. And if you have one heterozygous or homozygous for those amino acid substituting mutation, less than 1 microgram per ml. Now, this is the EM. We never published, but just so that I think you can have ideas. So that we usually call it like a bunch of tulips. It's a hexamer, and probably this one's much easier. You can appreciate the 3 of them. So when we started MBL, I've been working on this couple of decades, and only interest was infection. So the first report was the children who have recurrent infection had low or undetectable MBL. And soon afterwards, the life expectancy of cystic fibrosis patients was shorter in MBL deficient people. On the other hand, these MBL deficient people are better against intracellular pathogen. At the time, we knew only 12 TB. So because of this clinical notion, we generated MBL knockout mice. Now I have to mention gene compared to mouse and human. So mouse, they have 2 protein derived from totally different chromosomes, MBL-A and MBL-C. MBL-A is acute phase protein. So, and while MBL-C is consistently expressed. So when it's acute phase protein, so the naive mice, MBL-A is not really much, but once like LPS challenge, then MBL-A shoots up. Now compared to this, human, we have only 1 protein, which is genetically homologous to MBL-C. So often some people might describe MBL-C, human MBL-C. But, and if we compare the protein level, actually 40–50% homology between these 3 human, mouse, A, and C. So when I'll give you phenotypes, I'll talk about MBL knockout phenotypes. So MBL knockout mice are actually double knockout of MBL-A and MBL-C. So we made single knockout and cross to make double knockout. So MBL-no mice. So I'd like to mention, so there are, sounds very similar molecules out there now. First of it, it's LMAN1, which is also called ELG-IC53 or mannose receptor, which we also worked on. But I'm not going to talk anything about this, but both these sound cruelly similar molecules are on the membrane protein. And so when we made MBL mice, mice were healthy and fertile. So this was really great for us. And also we didn't see any obvious phenotype even at the histological level. And we made sure there's no transcript, no protein, and lectin complement pathway was undetectable. Most of the talk I'm going to share are already expressed. So if you would like to see detail, please read the original. Okay. And so first of phenotypes, we expected, yes, MBL mice are susceptible to the infection and impaired acute inflammatory responses. So I would like to share two cases. One, Staph aureus blood-borne infection. So we inoculated mice with Staph intravenously, and red column for MBL null and gray for wild type. So this data is bacterial titer 2 hours post inoculation. So you can see significant, at least 1 magnitude more bacterial titer in MBL knockout mice compared to wild type mice. And at this 2 hours, we also looked at cytokine TNF alpha IL-6. Both were diminished in MBL mice. So, and a second infection example was a viral infection, influenza A virus infection, and is the primary lung infection. And in this data, MBL mice are in closed bar and wild type in open bars. So day 1 post infection, still MBL knockout mice lungs could not clear a virus, while wild type almost kind of undetectable. And we also looked at, just part of inflammatory response, we looked at white blood cells and percent of neutrophils. You can see response, these white cell numbers and neutrophil populations in MBL lungs were different from wild type. And I was drawn to imaging all the time. And at this time, I had only FDG accessible. But I did chronological imaging post-infection day 1, day 4, and day 8. Left panel showing wild type mice and the right panel showing MBL null mice. So by day 8, there was almost no signal in the wild type mice lungs, but you can see some activity, FDG uptake activity in MBL null mice. So supporting again, MBL null mice have aberrant response to inflammatory response. Now, based on the MBL pathogen, broad pathogen recognition, there is a blood cleansing. It's like a dialysis, but the MBL cartridge is incorporated. So the idea is to clean pathogen from blood stream. It's early stage of clinical trial. And now from here, I'd like to share with you the phenotype we didn't expect, because we didn't really expect much actually. But surprisingly, first of all, MBL null mice responded greatly to polysaccharide vaccine. And so we had a collaborator at the time working on group-B strep type 3 polysaccharide, which was conjugated tetanus toxoid. And altogether, we immunized 3 times, 20 days apart. And this IgG titers are measured at day 60. And this data shows you group-B strep polysaccharide 3 specific IgG. We also measure just the whole total IgG, IgM. We didn't see a difference between wild type and MDL. So I'd like you to focus on between wild type and MDL, and increased IgG isotype was IgG1. And then this increase was in part increased total peritoneal cell number. So peritoneum, that's where we immunized. We used to immunize. And B1 cell was significantly increased in MDL normal mice. And so the next phenotype was coagulopathy. So MDL null mice had a prolonged bleeding time. And now in mice, you can do kind of simple experiments. So you cut 2 millimeter tip of a tail and put in 37 warm PBS and time till you stop seeing the blood is coming into PBS. So most wild type stops less than 1 minute. While MDL normal mice, many of them didn't stop even 15 minutes. And at this time, we wanted to make sure, the other coagulation factors are not altered in these mice. And as far as we looked at, like platelets numbers and platelet functions were comparable to wild type. And more so when we pretreated these MDL null mice with recombinant human MDL, the phenotype went back. So we could say this prolonged bleeding time was due to lack of MDL. And C3, now by the time we had a mice, people start saying, oh, MDL is a complement protein, but this result shows you this prolonged bleeding time was C3 independent. Now, as I mentioned earlier, MDL forms a complex with MASP1. And so we subjected to the same experiment, MASP1 knockout, MASP1 null, and MASP2 null. I'm saying MASP1 null, but actually, so what MASP1 null is MASP1 3 double null, and MASP2 null is MASP2 and SMAP null. So as you can see, MASP1 null mice had prolonged bleeding time, but not MASP2. So we conclude that this prolonged bleeding time coagulopathy was due to lack of MBL and or MASP1. And this work was done by Peter Gall's group. So MASP is directly linked to pro-inflammatory pathway. So here is the MBL's target and MBL MASP complex and PAR, protease activated receptor. There are 4 of them nowadays. And so what happened is when this complex binds to the target, MBL target, MASP can activate PAR and seems to like PAR2. By the way, PAR1 was also known as thrombin receptor. And this PAR activation actually directly linked to pro-inflammatory cytokine signaling pathway. So this is a link between MDL being acute phase and at the molecular level. And the next was bound phenotype. So we used dry bound, so we bound the back of mice. And in the wild type, what happened is ESCA or the burnt skin. slow fall overnight. But it was not, oh, this is a picture. So it's kind of expensive, but I was told that mice don't really feel pain. Well, any case, so the wild type ESCA shed off, so you can see muscle, back muscle, while MBL no mice didn't. And so most 21 out of 23 wild type lost ESCA like this, but only 3 out of 18. And in other words, 15 out of 18 had kind of looking intact skin. And once again, this phenotype was reversed or corrected by adding pretreating MBL no mice with recombinant human MBL. And again, C3 complement 3 or complement 4, complement 1Q knockout mice were more like a comparable to white type mice. And we wanted to see what's going on. So we did histological analysis and kind of adjacent skin to the band area. And one of which we did was to look at MMP. We used fluorogenic substrate for collagenase and gelatinase. You can see this green area. Those are MMP active, while we didn't really see much in MBL no mice. And more surprising was MBL no mice, the skin got almost like a 2 or 3 times thicker overnight. And we didn't really do anything further. Now, many of these experiments were done using anywhere between 8 and 12 weeks mice. Now, we wanted to know if there's any age-related issue. So we aged them average like 20 months. And what we saw was nausea, non-alcohol liver steatohepatitis, or simply fatty liver. And when we sent organs to histology, we sent out. And one of the things they noticed was fatty change, meaning the color of liver was actually different. And so we looked at, we did oil red or staining, which stains lipid. So this is wild type and 20 months old. You can see here and there this orange color that means the lipid droplets. In contrast, we saw two types of lipid deposit, in small deposits all over, or a huge lipid oil red or staining. And we consulted pathology, and they said they see similar pattern in human as well. And we also looked at fatty acid analysis, and 20 out of 23, I think 16 and 18, we had only 3 each, white type and MBL knockout mice, but 16 and 18 change seems to be higher in MBL knockout. Now, we never published it. We couldn't find any collaborator nearby. So, but we noticed the MBL no mice are tame and very sensitive to any stress. I know this characteristic is the most relevant to this meeting, but we haven't really done anything. The one thing maybe close, or at least something happened in the brain is one collaborator at the time was doing traumatic brain injury in mice. So MBL no mice had a slow recovery. So, this was just only in vitro. Lastly, this probably relate to brain house cleaning. So this was just like an ELISA type simple binding assay, but amyloid beta 40, 42, and this is just as a kind of control for the experiment. And red cross bars shows you their binding as 100%. And interestingly, the sulfate sugars inhibited the binding. to a data. And based on this results, even though people didn't really think this system-rich domain as a binding function, it's most likely this system-rich domain, not just bundle make form oligomer, but functioning as a binding domain. And so I'd like to believe that Mdl probably working to maintain balance of amyloid beta. Now we know our brain seems to need amyloid beta to brain health, I believe. So I think somehow Mdl participate sort of orchestrating whole innate immune or homeostasis somehow. And so the summary of the Mdl effects. So the Mdl status is unnoticeable in general. You wouldn't know, you don't feel it. But it might become an issue if you have underlying conditions and some infections. And it can be 3 ways. Of course, you know, just healthy, nothing. But too little or even too much seems to be not really good. And when I started, I showed you earlier, we had only a few clinical observations, but now the list has been expanding, and autoimmune diseases and also Crohn's disease and some neuronal programs. And also when we started, only deficiency was kind of big deal, but now we know too much is not really good either. Now, I'd like to touch a bit what I'm primarily involved with is the development of PET tracer. To me, seeing is believing, and but pain-free kind of slow development for PET tracer actually. Now, one of which successful is tracer is developed by Dr. Brugaloras, and it's on a phase 1 clinical trial, which is 3F4AP. It's a derivative of MS drug for amylopyridine. So this is a cation of a neuron. Neuron is covered or protected by myelin sheath. But if there is any damage, including MS, could be stroke or even Alzheimer, this myelin sheath is damaged and exposed potassium channel, voltage gated potassium channel became exposed, and this tracer can bind. And so this is one, this is, yeah, published. So the, sorry. So we were lucky, I'm sorry, but monkey had an accidental brain trauma injury at this area. And compared to other sort of commonly used tracer, PBR28 or PIB or FTG, CF4AP picked really more strongly. And so the currently under production studies, these tracers are made by radiochemists, and metabolic related receptors and some inflammatory related molecules, and also pain. And we hope that the tracer, if we hit the right tracer, we could provide early non-invasive and objective diagnostics, and possibly therapy too, depends on what we are doing. And lastly, the acknowledgement, I'm member of a Golden Center for Medical Imaging, and particularly thank you to George El-Fakri, he's the director, and my collaborators, Pedro Bugarotas and Norman Dean, and many people are sort of helped me to study Mdl project, and Alan Zuckerts, who cloned Mdl both human and mouse, if I didn't meet him, I would not be working on MDL and Mike Super and so forth. Thank you very much.
And thank you, Kazu. We are short on time, not your fault, because you also inherited a little bit of a delay there as well. But what I think we should do is probably move to break until 11:25. So it's a short 7-minute break. I think we all need a little bit time to stretch our legs. But I would encourage everyone to put your questions for Kazu in the Q&A. I know I have a question, which I'll enter into the Q&A. That way, at least we'll both be able to have a break. And